Hello to our friends joining us via recording. We are going to wrap up lesson number 4 today, and we're hopefully going to have a little bit of time to review for exam number 1. We started our class today by hashing out some statements about the types of transport that we looked at in class yesterday. So in class yesterday, we primarily focused on the 2 types of active transport, primary active transport and secondary active transport. What we talked about a couple of days ago was facilitated diffusion. So we're trying to figure out which of those types of transport apply to each of our statements that we have listed here. Before, I want to make sure we get a definition here on our slide. Help me out in the chat for my friends here today. When I talk about facilitated diffusion, what does that mean? How would you describe facilitated diffusion? I want to make sure we know what facilitated diffusion is. Okay, so Gloria's mentioning we use a protein. Yep, we use a protein to move things. Yeah, and Jacqueline's saying 100% correct that it's just like regular diffusion, just like regular diffusion. So the 2 things we should know with our facilitated diffusion, we are doing regular diffusion, so it's passive transport. We're always going from high to low, but we have to use the protein to make it happen. And the reason we have to use a protein, like we've got here in the chat, is remember there are some things that can't diffuse across the membrane. There's some things that the middle of the membrane says, nope, not letting you through. But if that thing needs to get in, we have to use a protein. We have to make a special channel for it. So absolutely, some of the things that we listed here, we would use this for things that are polar, for things that are hydrophilic. The other word that we've used that means the same thing as all of these things is water soluble. Oops, water soluble. Oh, yep, ions as well. Thank you, Nicole. Man, forgetting about those is terrible on this day because we're going to talk a lot about ions moving today. So big idea being anything that can't get across the membrane on its own, I have to use a protein to do it. If I'm going from high to low, I would call that facilitated diffusion. If I'm going from low to high, if I'm going up my concentration gradient, that's going to require energy. So our first statement here asked about energy. Both of my types of active transport require energy of some kind. Talking about doing active transport, we are, and this will come up later, right next to it actually, we're going from low concentration to high concentration. If I'm going to go from low concentration to high concentration, I require energy. So primary active transport, secondary active transport. They're active, energy is required. Please make sure we've written this down. Let's start it. Facilitated diffusion does not require energy. Facilitated diffusion does not require energy. No energy required because I'm going from high concentration to low concentration. That never takes me energy. I might have to use a protein to make it happen because I can't send it through the membrane, but I never have to use energy to go from high concentration to low concentration. So facilitated diffusion never takes energy. Active transport always takes energy. And the reason it takes energy, we'll bump over to our statement over here, is because like we said, we're going up the concentration gradient. Meaning going from somewhere where there's a lower concentration to somewhere where there's a higher concentration. If we just think about this physically, from low elevation to high elevation, that's going up. So both of my types of active transport, we move things up their concentration gradient. Now my statement over here talks about transporting things from high concentration to low concentration. That means I'm going down my concentration gradient. So if I go down my concentration gradient, I'm doing passive transport. My only example of passive transport on that list of types is diffusion. So the only statement that this applies to is facilitated diffusion. Now, I'm going to keep that in the back of my mind because we had a good chat as a class. But overall, in this statement, moving things from high to low, that's generally the job of facilitated diffusion. Here's a question that gave us a little trouble, right? We got our little question mark here. When we talk about using a protein to transport things, we ended up on, as a group, all three types. And that is correct. It is correct that all three types of the transport that we talked about in the last couple of days in class, all three of them use proteins. Now, when we're talking about facilitated diffusion, the job of this protein is just to make a channel, to make a passageway for stuff to sneak into or out of the cell. It's still no energy, but it is a protein channel to get us there. When we talk about primary active transport or secondary active transport, the reason that I have to use a tunnel is not only because I've got something that can't get through on its own, but it's also because I'm in some form to move it. So in these active transport processes, it still takes me, oh, so I'm breaking out a little bit. I'm plugged in today too. I don't know why it's doing that. Come on, friend. Let's see. Trying to make my internet better here. Bear with me, it might cut me out, but let me try something really fast. Am I back? Am I still breaking up? Can we hear me? Maybe we can't hear me. Okay, we can hear me now. Hopefully, record's giving me trouble today. Sorry. Somebody help me out in the chat. What was the last thing you heard? If we remember. 30 seconds ago. That's a long time, right? Of course, the most important thing is, okay, something about proteins. Okay. Well, so we were chatting about this one right here. So using proteins to transport things. All three types do use proteins. All three types of this kind of transport are moving things that can't sneak across the membrane. So all of them have to use proteins. The extra reason they have to use a protein is because they need something for the energy to work on. So for primary active transport, like we identified down here, we're using the energy of ATP to change the shape of our protein and help it to move that molecule from where there's not very much of it to where there's already a lot. So I use this ATP to change the shape of my protein in primary active transport in a way that helps me to get things across the membrane. When I talk about secondary active transport, I use the energy of, and it's kind of weird, but this is the way it's worded in the lesson and in your packet, the concentration of another molecule. When I do secondary active transport, the energy comes from one molecule that goes from a place where there's a high concentration, it goes downhill to where there's a low concentration. That allows the other molecule to go uphill. So from a place where there's not a lot to a place where there's already a lot. We did in class yesterday that boulder hill example where we rolled things down the hill to knock something else up the hill. That's secondary active transport. When somebody goes from high to low to allow somebody else to go from low to high. The only way it's possible for me to do active transport a protein that makes it happen. Somebody's got to harness that energy that makes it active transport. Otherwise, I can't do it. So anytime we're talking about active transport, gotta have a protein. When I'm talking about facilitated diffusion, gotta have a protein. When I'm talking about regular diffusion does or does not use a protein. Does regular diffusion use a protein? Yeah, it does not, like we're saying in the chat. Regular diffusion, or when you see the wording on a homework question or on an exam question, when I tell you something diffuses straight through the membrane, no protein required. If we can diffuse straight through the membrane, no protein required. Hey, let's remind ourselves, because this isn't a thing we've got to know for the exam next week. What are those words that we use to describe things that can diffuse straight through the membrane? What are those, to use our word, biochemical properties of things that can sneak through? Let's see what we got. Yep, we've got the word nonpolar. Nonpolar things can diffuse straight through. We've got the word hydrophobic. Hey, by the way, hydrophobic, how do I feel about water if I'm hydrophobic? Do I like it or I don't like it? Yeah, a couple of us are saying we're afraid of it, we hate it, don't like it. Absolutely. So nonpolar things, which means the same thing as hydrophobic, which also means the same thing. A couple of us typed this in as well. Our other word that we can say too is lipid soluble. If I'm nonpolar, or if I'm hydrophobic, or if I'm lipid soluble, I don't need a protein to get across the membrane. I can sneak straight through. That's why these kinds of things can do diffusion. These kinds of things that we listed over here on the left, things that are polar, things that are hydrophilic or water soluble, those kind of things can't do regular diffusion. They have to do facilitated diffusion. They have to go through a protein. So when we talk about any of these kind of things right here, something nonpolar, something hydrophobic, something lipid soluble, those don't need a transport protein. Everything else does need that protein because it doesn't match the middle of the membrane. When we talk about our types of transport, we gave two specific examples. Yesterday we talked about one of our types of transport that helps us to feed the cell, or it helps us to bring in what the cell really likes to eat. Help me out in the chat. I know I heard us discussing this. What's the cell's favorite food? What does it like to eat? Yeah, so the thing that a cell most likes to eat is glucose. That's its favorite. That's the easiest thing for it to break. So when we talk about how we get glucose into the cell, we use something called the sodium glucose cotransporter, and that was an example of secondary active transport. So secondary active transport, the specific example we gave was, here I'll type its name, the sodium glucose cotransporter. That was our specific example of secondary active transport. So that's why this statement applies to that. When we talk about polarizing the membrane, for polarizing the membrane was the sodium potassium pump. That was our specific example of primary active transport. And remind me again, when I do primary active, where does the energy come from? How do I make primary active transport work? Yeah, Gloria is correct. Primary active transport, I use the energy of ATP. We talked about it yesterday, kind of like energy monies. I use the cell's money, ATP, to move things in. I'm looking at the question here in the chat. Jacqueline says she's a little confused on why sodium moves out of the cell and why potassium moves into the cell when there's already a high concentration of those things? Yeah, so Jacqueline, that's why it's primary active transport, right? Because they're both moving to where they're already more concentrated. The reason that I move them that way is because of this right here. I move them the wrong ways, basically, if we're talking about how they want to go. I move them the wrong way because when I move enough of them the wrong way, my membrane gets polarized. Help me out in the chat. What does it mean again if the membrane is polarized? What does that polarized word mean? Yeah, so Nicole's identifying we're got different concentrations, different charges. Basically, the idea is things are different inside compared to outside. Yeah, Gloria said, sorry about the caps lock. I was like, wow, Gloria's really excited that she knows what polarized means. I mean, it's really important. So I'm glad that we know what polarized means. Yeah, so polarized means we're different inside and outside. We have to be different inside and outside for the cell to be alive. And today we're especially going to talk about how neurons work. The way that neurons work 100% depends on them being polarized. So we've got to get a different concentration of sodium outside compared to the potassium that's inside for us to be able to change the charge on the membrane. All right, we've talked about each of these statements. What questions do we still have about this? Any particular questions about this? Or give me an emoji. How are we feeling? Like I said, Gloria is really excited about polarized cells. I love it. Need to find an emoji that looks like polarized. Let's see if I can come up with something really fast. Okay, totally not polarized, but this is so cute. Look at that. I love it. I am partial to penguins, but that owl is pretty awesome. Oh yeah, okay. So Jacqueline did bring up a good question. I forgot to go back to that. So the question is, when we talk about secondary active transport, we're using the concentration gradient of one molecule to move another. So in secondary active transport, let's draw some lines. Somebody goes from high to low. That allows somebody else low to high. So when we talk about secondary active transport, could technically group it with both of these statements, right? Because both things happen in secondary active transport. So what we need to make sure we know about secondary active transport is one of the things that we're moving with it is going from high concentration to low concentration. That's giving the energy for the other thing to go from low to high or to go up. So this is why I purposefully put things like this on the stuff we talk about together. So you could put secondary active transport with a little star, with an asterisk to remind yourself when we talk about things going up, one thing that moves, one thing does go up, that's correct, but the other thing actually goes down. So we could put secondary active transport again with an asterisk over here on high to low, because the way that secondary active transport works, somebody goes down their concentration gradient so somebody else can go up. So yeah, great question. I'm glad you reminded me about that, Jacqueline. And she had mentioned earlier when we were discussing things, check that out on the homework assignment, because I do ask you, do both things move up their concentration gradient? Does one thing move up and one thing move down? Do both things move down? So check out that wording on the homework assignment because it is different. Transport, I do one of each. In primary active transport, both of them go up. In facilitated diffusion, we only ever go down. Because remember, it's diffusion. It's passive transport. Yeah, so thank you for that reminder. I'm glad we were able to mention that too. All right, if there are no other questions about this, I want to understand why we were talking about this kind of stuff. The reason we were talking about this kind of stuff is because of neurons. So again, we're going to talk more about neurons in unit number 3, but we're going to introduce them here in unit number 1 because we just talked about all of the types of transport proteins that are important for how neurons work. So when I talk about neurons, there's this term that I do want you to be familiar with. I'll relate it to another term for us. It's called resting membrane potential. Resting membrane potential. We're going to break this down a little bit. This word potential right here, whenever you see potential, I want you to be thinking charge, as in an electrical charge. So whenever we see the word potential, we should be changing that in our mind to charge. Resting membrane charge. When that word comes back, that is polarized, a cell that is polarized, it has a potential. It has a different charge on the inside compared to the outside. Ugh, so sorry, I cut out again. Am I back or am I still choppy? I know, online life, right? Okay, we're good for now. Okay, so let me just reiterate what I said before. If we have, it comes in and out, yeah, okay. In a bit, I might try one more last-ditch effort, and it might go poorly, we'll see. When we talk about a cell that is polarized, that means that it has a resting membrane potential. It means that the inside is different from the outside. In pretty much all the cells in your body, except for muscles. So pretty much everybody, the number that I want you guys to know is this number right here, -70. Underline, highlight, star that. I'm about 95% certain I will ask you for that number on the exam. So -70 millivolts, that's my resting membrane potential. So that means that in normal circumstances, when a neuron's doing nothing, resting membrane potential. When a neuron is waiting to hear from its neighbor. So the inside has lots of negatives inside of it. The outside has lots of positives. When I compare those two sides, we're different. And remind me in the chat, polarized is good or bad? If a cell is polarized, is that good or bad? Yeah, that's good. It's good for a cell to be polarized. We want it to be different on the inside, -70 on the inside, compared to. Okay, this is probably Dr. Ollis's favorite graph of the whole unit 3. We're going to use this graph to death in unit number 3, but we're going to start talking about it now. Oh my gosh, I keep cutting out. Okay. We're going to take a drastic measure here, and I am going to turn the Wi-Fi off on my computer to try to make it use my cord. There's a decent chance it's going to drop me from the room, so I promise I'm not leaving. Stick around. Let me see if I can get it to just use my plug. Let's see. Here goes nothing. Okay, I tried to get my computer to do the plugin and my computer is really not happy. So I apologize. I'm probably going to keep dropping today, but if I do, keep showing me in the chat that I dropped and I will go back and repeat myself. So I apologize. I am having my computer's like, it's the end of the week. Why are we still working? So I apologize. It looks like the video on my end says it's still recording, so I'll see what happens with the recording. If it gives me 2, I'll make sure to post both of them. If it just gives me 1, then that would be even better. So we'll see. Yeah, I could try a hotspot. I feel like that would be even worse, but it's worth a shot if I keep having all these problems. So sorry again, guys. Let me mention before we, so I'm gonna go back to my slide here. When we talk about resting membrane potential, so the normal charge on the membrane, when I say that it's -70, what that means is that inside I basically have 70 more negatives than I have on the outside. So if I was measuring negatives versus positives, I've got a whole lot more ions with a positive charge outside compared to my ions on the inside. Because I have so many more positives outside than I have inside, that's why the charge is going to read it as -70. So we don't really, we're basically when the question asks compared to what, we're comparing the inside to the outside. The inside looks a lot more negative. The outside looks a lot more positive because of those ions with their charges. And we also have inside the cell a lot of proteins that have a negative charge on them too. So there's a whole lot of negativity inside the cell compared to the outside of the cell. Does that make more sense to that question, what we're comparing? Okay, yeah, so it's just a comparison. If I looked at the soup inside here, it would look more negative than the soup that's up here. This looks more positive. Hey, let's do our reviews because it'll show up on the exam. What's the name of a cell? Intracellular fluid. What's that special name for the soup inside a cell? Yep, that soup inside the cell is cytosol. Oops, cytosol. And let's say that this is a cell that's inside a blood vessel. What would the name of the extracellular fluid be? If we're inside a blood vessel, but we're outside the cells, blood vessels are filled with, with plasma. Plasma, perfect. Yeah, Nicole said at least I'll get that question right. Yep, there's going to be some gimmies. There'll be some gimmies, so find them because it'll make you feel better, right? And if we're inside a tissue, not inside a blood vessel, if we're inside tissues, what's that extracellular fluid out there? Inside tissues, what's that one called? That's interstitial fluid. Absolutely. Okay, so this resting membrane potential thing means that cytosol is -70 millivolts. It's 70 millivolts more negative than plasma is. Plasma is more positive, or interstitial fluid is more positive than cytosol is. So a good comparison with some reviews for us to review those big words. Yeah, Jacqueline says a few questions will be guaranteed on the exam. Absolutely. Oh yeah, there's going to be some questions that I know that you guys know. And I always tell my students too, when you're taking the exam, if you hit a question that you're like, I have no idea what the answer is, come back to it later. Like, go find a question that you do know the answer to, because there's going to be more than just a few. There's going to be several that you definitely know the answer to. So go find those ones that you definitely back later to those ones that are giving you trouble. Okay, so before I died and my internet was revolting against me, I told you that this is my favorite graph from unit number 3, just because we're going to understand it really well. We're going to start right now by learning it and getting a little bit of an idea of what's happening. But by the end of unit 3, you're going to be able to draw this graph. Let's start with the two edges of this graph. So over here on the left, we have a time period that says at rest. Over here on the right, we have a time period that says we're returning to rest. When we see this rest thing here, that should trigger alarm bells in our mind that we are doing that resting membrane potential. So the two parts on this graph, sorry, I'm typing it in over here too. The two parts on this graph that are mentioning rest, we are at resting membrane potential. Who caught it from before? What was that easy word for potential? What's the other thing that you can say potential means? What are we putting instead? Yeah, we're putting charge. We're putting charge there instead. Okay, so when a neuron is at resting membrane potential, it's not doing anything. Its charge is normal. It's chilling out. It's listening to hear it talk to it. Now, the way that neurons talk to each other is they use a compound called neurotransmitters. When I look right here at this part of the graph called stimulus applied, point number 2. Stimulus applied, point number 2, is when one neuron talks to another neuron by spitting out neurotransmitters. So one neuron gives a neurotransmitter to the other neuron, and when we send neurotransmitters to each other, those neurotransmitters attach to a particular type of gated channel. When I'm talking about a neurotransmitter, does anyone know which of our types of gated channels a neurotransmitter would attach to? Yeah, so a couple of us are chiming in. Neurotransmitters are a type of chemical. So they're going to attach to chemically gated channels. Chemically gated channels. Now, here's a reminder for us that all of my gated channels help me to do facilitated diffusion. That would be a underline highlight star idea for you. All of my kinds of gated channels, chemically gated channels, voltage gated channels, mechanically gated channels, all of my channels help me to do facilitated diffusion. Facilitated diffusion, do I go from high to low or low to high? In facilitated diffusion, which way do things move? Yeah, they go from high to low because it's no energy required. Okay, so in particular, the type of channels that get activated by these neurotransmitters, when we're looking at this graph, the particular kind of chemically gated channels that are getting activated are chemically gated sodium channels. Chemically gated sodium channels. I abbreviated there, so infer with me. Chemically gated sodium channels. Hey, unit number 1, salty banana. Salty banana. Let's draw ourselves a little banana. Okay, let's make sure we know this. What do I have a lot of inside my salty banana cell? What are some of those things we said we had a lot of inside my banana? Okay, we all got that potassium. What else is inside? Okay, so a couple of us have mentioned proteins, yep, and a couple other of us have mentioned glucose. Inside my banana, potassium, glucose, proteins. 100% got to know that. It's a salty banana. What do I have on the outside? Lots of this stuff on the outside. Salty banana. Who's out there? Yep, sodium's out there. That's my first half. And yep, my second half is chloride. 100%, we got to know this. Guarantee that. Here's why it matters that we know that it's soup. Know the recipe for these soups. Chemically gated sodium channels. When I attach a neurotransmitter to them, I open up these channels, these chemically gated sodium channels. They're going to move sodium. They're moving sodium using facilitated diffusion. So they're moving it from where there's a high concentration to where there's a low concentration. When I open up a chemically gated sodium channel, which way does sodium want to move? In or out? Which way is sodium moving when I open up this channel? Exactly. Sodium wants to go in. We're doing facilitated diffusion, right? We can only go from where there's a lot of me to where there's a little bit of me. When I open up a chemically gated sodium channel, sodium rushes into the cell. Hey, sodium has a positive charge. So sodium is an ion with positive charge. Who remembers the name of an ion with a positive charge? What's the special name? Yeah, the special name is a cation, right? I always like to remember that by thinking of a cat with little pluses for eyes. A cation is an ion that has a positive charge. When we talk about chloride, that's an ion with a negative charge. That's called an anion. I'll type that in chat for us, an anion. Sodium is a cation. It has a positive charge. When I open up a channel, when I open up a chemically gated sodium channel, I'm making it possible for things with a positive charge to come into my cell. So see how we started at -70. Remember, that was one of those numbers we had to know. -70, resting membrane potential. When I let in a bunch of extra sodiums, because I opened a chemically gated sodium channel, sodium rushes in, it rushes in and in. Suddenly I've got a whole bunch of more positives that are inside my cell. So because sodium's a cation, and because it's more concentrated outside than inside, when I open up a channel for it, it rushes into the cell and makes the plasma membrane more positive. So the first kind of channel that gets activated on a neuron is called a chemically gated sodium channel. That allows sodium to rush into my cell. So first number we need to know, -70. This is when a neuron is at rest, when it's listening. When listening and it gets a neurotransmitter, when there is a message for it to hear, that message can open up these sodium channels. These sodium channels make its charge, make the inside of the membrane get more positive. Next number we have to know. Here's the next number we got to learn. -55. -55. -55 is a number that we call threshold. The reason that threshold matters is because when a neuron hits threshold, this is enough from normal that the neuron decides, I cut out. What was the last thing you heard? Can we hear me yet? Okay, threshold. Okay, well, we'll pick back up there. Is the charge that when a neuron hits it, when a neuron hits -55, when we hit this charge, the neuron knows that it's time for us to talk to our neighbors. So we hit threshold, we're at negative 55. We're different enough from normal. Our charge is different enough from normal that now I open up a different kind of channels. Which kind of gated channels open when charged? Or electricity? What kind of gated channels use charge? Voltage gated channels. When the charge gets wrong, in this case, when we get to negative 55, when we're 15 millivolts above where we're supposed to be, at that point, voltage gated sodium channels, should have put that word in there before, voltage gated sodium channels are going to open up. Voltage gated means my charge has to be different. If my charge gets all the way to negative 55 compared to normal, negative 70, if I get up to here, these channels say, okay, we're committed, we're in. Voltage gated sodium channels open up. Voltage gated sodium channels, there's a whole bunch more of them on a neuron. As soon as I open these, a ton of sodium rushes into the cell. And you can see what's happening to our charge here. All those positive sodiums are making it go up and up and up. We hit 0, where positive and negative inside versus outside is the same. But I keep bringing in more and more and more sodium until I get all the way up to positive 30. That's the last number I'm going to make you know. Positive 30. I bring in so much sodium with all of its positive 30. And at this point, the neuron realizes, oh no, I am completely flipped out. I was supposed to be negative 70. I'm positive 30. My charge is completely wrong. This charge being wrong is going to open up another kind of channel. This kind of channel opens because of a charge problem. Again, which kind of gates respond to charge problems? If my charge is wrong and it opens up a gated channel, yeah, it's still going to be voltage gated. But this time, when we're all the way at positive 30, I don't need a voltage gated sodium channel to open. At this point, I'm going to open a voltage gated potassium channel. Voltage gated potassium channel is going to move potassium using facilitated diffusion. When I open a voltage gated potassium channel, which way is potassium going to move? Where's potassium going to go if I open up a channel for it? Yeah, potassium's going to go down its concentration gradient. I've got a lot of it inside. Potassium is going to leave. Potassium's going to leave when I open up a channel for it. Potassium is still a cation. It still has a positive charge. When potassium with its positive charge is leaving, think about it kind of like we're doing subtraction. So I have a positive one that leaves, so -1, -1, -1, -1. I'm spitting out all of these positives, all of these potassiums, they're all going out. As potassium leaves the cell, the charge on the inside gets more negative because all my positives, in fact, so many of my positives go away that I actually end up all the way down here where I'm actually more negative than normal, more negative than I should be, because I've spit out so many of my positives. I lost so many potassiums. At this point, to help me get back to normal, I use a couple of different things. Let me draw a little arrow for us here. A couple of different things help us get back to normal. One of those is that primary active transport example that we did, the sodium potassium pump. That's one of the ways I get back to normal. The other way I get back to normal, though, are a special type of channels called leakage channels. Leakage channels. I'm forgetful. I need your help. Leakage channels are or are not always open. Leakage channels are or not always open. Yeah, they are. Yep. So leakage channels are always open. Always transport stuff from high concentration to low concentration. So when I get too negative down here, one of the slow ways that I work on getting back up to positive is using those leakage channels. They start drip, drip, dripping my potassiums back into the cell because I just spit them all out. They spit out a few of those sodiums, get things back to normal. But we also use that sodium potassium pump to help us pump out sodiums and pump potassiums back in. That's going to go more quickly than just those leakage channels. I just talked you through the entire graph here, talked a lot, so I need you to help me know what questions we have. So Nicole said she thought the graph was a sodium potassium pump. It is not. The sodium potassium pump, all that does is it at the same time moves sodium in, or excuse me, moves sodium out and moves potassium in. When we look at this graph, the only part that we see the sodium potassium pump being part of is this very end part right here. Pretty much everything we're seeing on this graph is actually these gated channels, the gated channels that we talked about at the beginning of the lesson. So gated channels are what neurons use for the most part to help them change their charge from being not polarized, or from being polarized to depolarized, the wrong charge, back to normal. Yeah, a couple of us are saying in the chat, I'm going to watch this video many, many times. That's a good idea. Honestly, so here, if you want a picture of this, take a screenshot. It'll be in the video. I'm going to clear it off, and I'm going to show you how I think you should study it. So take your pictures really fast if you want one. Here comes the eraser. What I think you should do to study this graph is for each of these parts of the graph, you should ask yourself what kind of ion channels are open. For each part of the graph, I think you should ask yourself what kind of ion channels are open, or what opens here. At this part on my graph right here, the neuron received a stimulus, it got a message, and that message was a chemical. So the type of channels that are open right here are my chemically gated sodium channels, because I use a chemical to open the gate right here. When I open enough chemically gated channels that open with chemicals, I'm going to change my charge on the membrane all the way up to -55. If my membrane charge gets different enough, now I open up voltage gated channels because my charge has changed. Once my charge changes, those channels go nuts and they move in all of the sodium because they end up getting us all the way positive. And again, we have a problem with our charge. Since the charge is wrong, it's a voltage gated channel that's going to open. And that voltage gated channel helps to get us back to normal by spitting out our potassiums. So we want to know what kind of gated channels are being activated at each of these places. For right now, I'd love for you to understand it, but if we don't have time to understand it before the exam, at least memorize it. So if we're going to memorize it, voltage-gated potassium on the right. On the left, we've got voltage-gated sodium. At this point right here, we've got chemically gated sodium. Like I said, if we don't have time to understand it right now, totally fine. We just need to at least know for sure what's open where. Jacqueline asked, there is only chemically gated sodium, no chemically gated potassium. Unfortunately, that's incorrect. But for the sake of our lesson right now, the only kind of chemically gated channels we care about in Unit 1 are chemically gated sodium channels. Because see, here's the deal. Let's draw our cell again. Our cell, the ones we're talking about, are sodium, sodium and potassium. If I open up a chemically gated potassium channel, which way is potassium going to move? Which way is potassium going to move if I open up a channel for it? Yeah, potassium's going to go out, right? Potassium is always going to leave a cell. It doesn't matter if I use a voltage-gated channel or a chemically gated channel to make it leave the cell. Potassium always leaves. So when I'm looking at this part of the graph, if this was a chemically gated potassium channel, I would be out potassiums, spits out positives, which would actually make the charge on my membrane go negative. So we would see on this graph, and we'll see this in Unit 3, you can actually make the line go down lower. If my line goes down lower, that's what happens when we have a chemically gated potassium channel, chemically gated potassium. We're not caring about chemically gated potassium right now. Right now, all we're caring about is chemically gated sodium. So chemically gated sodium, voltage-gated sodium, voltage-gated potassium, and then those leakage channels and that pump. Let me bounce back up. Okay, where does depolarization and repolarization happen? So depolarization is the process of making things more positive. Depolarization equals becoming more positive. Depolarization. Repolarization is the process that I use to become more negative. So when I look at this graph, anything that makes me more positive, that's depolarization. Everything that's making me more negative to get me back to normal, that's repolarization. So at time 0, I am, or maybe the 0 we're referring to actually is this 0 right here. At the very beginning and at the very end, we're always at rest. Anything that makes the charge go up is always called depolarization. Anything that makes the charge go down is repolarization. So it doesn't, 0, honestly, when you're thinking about neurons, you should actually kind of ignore this number 0. 0 doesn't matter to us. We blaze right through it, going all the way up to positive 30, or we blaze right through it when we're going back down, when we're repolarizing. So 0, not a big deal. We don't care about 0. Is the voltage-gated potassium what we use to help the cell return to resting membrane potential? Yes, that's correct. Voltage-gated potassium channels, their goal is to try to get me back to normal. Voltage-gated sodium channels, they take me away from normal. They help with depolarization. So voltage-gated potassium channels do repolarization. Voltage-gated sodium channels do depolarization. Yeah, so Jacqueline asked, does depolarized mean we're making them the same? Yeah, we're going from, if they were completely the same, it would be right here at 0. We go from being very different, meaning inside's negative, all the way up to 0, all the way up to the inside suddenly being more positive. And that's bad news for my neuron. The neuron does not like to be positive on the inside. So it's going to freak back out or flip back out down to being negative again. Yes, so Gloria asked about voltage-gated channels opening at positive 30. Yep, in particular, the type of voltage-gated channels that open at positive 30 are voltage-gated potassium channels. When I get all the way up to positive 30, I don't need to go any more positive. That is as positive as a neuron is going to get. It's so positive that it realizes it needs to go back negative. And the only way it can go back negative is using these potassium channels to make it more negative. So chemically gated the chemically gated makes it more positive. Chemically gated sodium channels, yes, they make the membrane charge more positive. When I get to this charge, negative 55, when I get that positive, when I've moved that much, at that point, my voltage gated channels open up. They like I said, they said, oh, great, we really want to go positive because we're all the way at negative 55. Because we're at a different charge compared to normal, I'll open up those voltage gated sodium channels. What other questions, thoughts, emojis do we have? Yeah, so Kai asked a clarifying question. At negative 55, that is correct, that at negative 55 is when voltage-gated sodium channels open up. The charge or the voltage that makes this type of channel open is negative 55. When I get all the way up to there, those guys open up. When I get all the way up to positive 30, the voltage of positive 30, that's what's going to cause voltage-gated potassium channels to open up. So they open up at one voltage, positive 30. My voltage-gated sodium channels open up at a different voltage, at negative 55. Let me, oh, thank you, Jacqueline, that's sweet. Let me share a whiteboard with you because I want to, so this graph, I know that it's slightly terrifying. Totally get that. Here's the big ideas I want us to get off of this graph right now. One of the ideas that we have to know to interpret this graph. First idea we have to know is how do different kinds of gated channels open? That's the first thing we need to know to understand this graph. How do different kinds of gated channels open? So let's help each other with this. We had some chemically gated channels. How do chemically gated channels open? What opens those channels up if they're chemically gated? Yeah, so our specific example is neurotransmitters. That's our specific example. It's just any kind of chemicals. Neurotransmitters are really the chemical that we use a lot. Okay, first kind of channels that we have are chemically gated channels. Second kind of channels that we have are voltage gated channels. Voltage gated channels. What causes a voltage gated channel to open? How does that kind of channel open up? Yeah, it's all about the charge, right? So the charge changes. That's what causes a voltage gated channel to open. Okay, here's thing number one that we need to know to be able to understand this graph. How do our different kinds of gates open? Thing number two that we have to know, well, here, we'll put it a different way. What is the recipe for cytosol versus interstitial, spelled that wrong, sorry. Second thing we need to know to understand that graph is our salty banana. So you all are going to hate it. I'm going to make it again. We're going to make another banana. Okay, here's my banana cell. Do we want to start on the inside or the outside? You guys get to pick. Inside or outside? Okay, Gloria wants to start inside. Here we go. What do we got inside? Tell me again. I always ask this. We will know this answer, right? When we get to the exam, we will know. Yep, okay. We got a whole lot of potassium. That's the big one. Glucose. Got a lot of proteins. Okay, that's inside. So what do we got going on outside? What do I have a lot of outside my cell? Whole lot of sodium, whole lot of chloride. Okay, second thing we need to know to understand what's going on with this graph is this recipe. What do we have a lot of on the inside? In particular, we're caring about potassium right now. What do we have a lot of on the outside? Sodium in particular is what we're caring about right now. Then we need to know, we'll add our next area here. We need to know what happens in facilitated diffusion. That's the next thing we need to know, because these gated channels are doing facilitated diffusion. So facilitated diffusion, we'll put our notes that we've already said before. This is passive transport, no energy required. This is going from high concentration to low concentration. This is the kind of transport that those gated proteins that we're talking about here, both of these types of gated channels do passive transport. So these three components, if we know these three things separately, these are the things that help us to understand the graph. So let me wing it here. Here's what that graph looks like. I've done this enough times to be able to draw it in my sleep, right? You'll get there, Unit 3. When I'm looking on this side of the graph, my membrane charge is going up. I need something to move in a direction that would make me more positive. So let's draw our directions on our graph here. If I open up a channel for potassium, remind me, is potassium going in or out? If I open up a channel for it, which way does potassium want to go? Okay, potassium wants to go out. There's a ton of it inside, right? It wants to go out. If I open up a channel for sodium, sodium wants to go which way? Yeah, sodium, there's a bunch of outside. Sodium wants to go in. When sodium moves inside, that makes my membrane charge more positive, because when I bring positives in, of course I cut out, right? Just when we're doing, it's like my computer knows when I'm saying important stuff. It's like, ha, sucker, you're not gonna hear this. Okay, am I back? I'm still cutting out, ugh. Okay, I might be back, maybe. I know it wants us to fail. I kind of feel like that's what technology says in general, right? It's like, you can't be this smart. I'm not going to let it happen. Okay, so here's what I was saying. Sodium moves into the cell. Sodium has a positive charge. When something with a positive charge comes into the cell, it makes the membrane more positive, because we're always comparing inside to outside. Potassium, it takes its positive with it to leave the cell. When potassium leaves the cell, it makes the membrane charge more negative. It makes the charge go down because we're stealing those positives from inside and we're sending them outside. So when you look at this graph and the membrane charge goes up, the only ion that we talk about moving that makes the charge more positive on the inside, it can only be sodium. Only sodium can make the membrane charge go up. And only potassium, when we're talking about sodium and potassium, only potassium, when it moves, makes the membrane charge go down, makes it more negative. So when you're looking at the graph, even if we're still struggling with our different types of gated channels, based on my salty banana and the way that things move, I can predict that all of these changes have to be due to sodium, some kind of gated sodium channel. The changes that I see over here have to be related to potassium, because potassium is what I can use to make that membrane charge go back down. So 3 big ideas that we learned separately are types of gated channels, what's going on with our ions, and how facilitated diffusion works. All of those things come together. I'm asking you to pull all 3 of those ideas together to understand how this graph. Make sure we understand each of these topics separately. When we understand these things, apply it to this graph and see if we can use that to help us understand it. That's how I would recommend breaking down this graph to understand the backstory, to know what's going on behind it. I am about out of time because I'm sharing my webcam again today, even though my computer's trying to kill us, right? It wants us to fail, as we said. Are there any quick last minute questions that we had for today? I will spend all of Monday. We will do exam review. Please feel free to email me questions this weekend, because I know we didn't really get a chance to do a lot of Unit 1 review stuff today. I apologize. So the question is the difference in the function of chemically gated sodium channels versus voltage gated sodium channels. Honestly, the function isn't much different. Both of them, their job is to move sodium into the cell. I suppose what we could say the difference would be is that chemically gated sodium channels, really their job is to help open voltage gated sodium channels. My voltage gated sodium channels, their job is to really rapidly depolarize the membrane. The voltage gated sodium channels work a little bit more quickly. So if we wanted to say a difference in their function, the voltage gated faster, they depolarize it faster. The chemically gated sodium channels, they've just got to get us to threshold. They've just got to get us to the point where these guys open up. Other than that, they both make the membrane positive, so their function is about the same. So the question asks about a homework question about carrier. Can you guys help me out in the chat? What was special about carrier proteins? How do carrier proteins work compared to channel proteins? What's different between a carrier and a channel? Yeah, so one of the big things is the carrier protein is going to change shape. It opens to the outside, and then, like Nicole mentioned, it attaches to what it's transporting, then it opens to the inside. So when we're talking about carrier proteins, it's a type of protein that has to be open to the outside to attach to something it's moving, and then it opens to the inside to release that. If you look at our pictures of the sodium glucose cotransporter and the sodium potassium pump, both of them, and then they open to the inside. They change their shape, they attach to what they're transporting. Nicole said I cut out. I was referring you to those pictures. Check out the pictures of sodium glucose cotransporter and sodium potassium pump. You'll see that they do change shapes. Ariel asked if there is unlimited attempts on the review assignment. Yes, and the review assignment is optional too, so it's not going to change your grade either way unless you happen to miss an assignment. If you missed something from unit number 1, I will manually go in later and replace a 0 with your score on that assignment. Otherwise, it's just there to help you review, and every time you do it, it's going to pull different questions. So please do it an unlimited number of times, because that'll help you bump into more of those questions. Unfortunately, I probably need to get off mic because I'm sharing the room. I'm going to stick around in the chat though, so if you have other questions, please feel free to do that. I did see the quick question about Proctorio. Yes, we are using it. So please make sure to install Proctorio on your computer. You can test to see if Proctorio is working by taking that practice test. It's not actually a test, it's just one question that asks, did you install Proctorio? There is not a password for it, so if your computer is asking for a password, Proctorio is not working on your computer. So click on that Secure Exam Proctor link and then go back and try to take the test again. That should activate Proctorio for you. So if it ever asks for a password, something's up with Proctorio. So make sure to get that installed. Make sure to click on that Secure Exam Proctor and then try to take the practice test. It should let you in there. Recording, turn off the mic, stick around for questions. Please feel free to email me this weekend. And 9:30 on Monday, we will do review together. Also, a quick plug again for supplemental instruction. Tomorrow at 9:30, Elise is going to do exam 1 review. If you are at all possibly available tomorrow morning at 9:30, plan to go to supplemental instruction. She's great. She will be a great help for you to help you prepare. So tomorrow at 9:30 and Monday at 9:30, we will review for that exam. So have a great weekend. Email me with any questions you have. Great to see you all today.